Or does it not?
They can see you on the camera.
Oh, okay.
Yeah.
So I'll be in and out.
Yeah.
Cool.
Go ahead.
Okay. Awesome. Thanks so much for that introduction. And thank you all for being here tonight on a weeknight at 7 p.m. I'm amazed by this group, and it's been super fun chatting with some of you so far. So as our speaker just alluded to, I'll be talking a little bit about native pollinators in our region. And I do preface apologies. Most of this is centered in California. I'll drop in a couple of tidbits pertaining to Nevada. But I'll be focusing.
Can I ask you to speak up?
Okay, I'll do my best. And I'll be speaking primarily about butterfly population change through time, some threats, as well as some stories of resilience, as I find is especially important, especially now, to focus on some of the areas where we are succeeding and where things are going correctly. Okay. Okay, so I'm Claire. I am part of Matt Forrester's lab here at the University of Nevada, Reno, where we focus on the ecology and evolution of butterflies. I'm particularly invested in the butterfly monitoring component of this lab, where I get to go out to a variety of beautiful, gorgeous, pristine sites within the Sierra Nevada mountain range. And we're also all heavily involved in conservation as well. And here are some of the other beautiful faces that are part of this lab. And of course, our fearless leader, Dr. Matt Forrester. So within this sphere, as I was discussing earlier, I am particularly interested in butterfly migrants beyond just the monarch. There are a number of other species that undergo some pretty amazing movements over the course of the summer season when they're flying, such as this goth butterfly here, Nymphalis antiopa, otherwise known as the morning cloak. This desert vagrant, Bifidum exilis, the western pygmy blue, it's actually the smallest butterfly in North America, and it can make this journey from the Mojave Desert all the way into Reno on especially good years where there's enough rain in the desert to produce enough food for this particular species. And finally, this lorquin that you'll see floating around, gliding gorgeously and effortlessly. For my talk today, I'll be focusing on the big picture and overall status of butterflies, just to give everybody a sense of where we stand. And then I'll get into some mechanisms. So some of the causes that are creating the patterns that we see, including climate change, pesticides, and a little bit of flowering plants, as there's a bit of a snippet of work that is just starting to come out now. Finally, rounding out with a couple of actions that are happening at the local level, as well as what we can do on the ground. So how on earth do we paint a picture of what's going on with butterflies in the American West? We have a variety of different landscape types, multiple different elevations, tons of different flora, and of course, our hundreds of species of butterflies that are flung over this entire landscape. Well, I'm going to be telling you a small story about 3 different data sets that were used to get an understanding and assessment of how butterflies are faring in this region. And the first of those is very near and dear to my heart, the Shapiro transects. So these transects are running along an elevational gradient within Northern California, beginning in the Central Valley and extending up into the Sierra Nevada. They were started by this amazing individual, Dr. Art Shapiro, in the 1970s, where he decided he was curious about butterfly phenology, that is the timing of life history events, selected these sites for their different land use types in that elevational gradient, as well as the fact that they're publicly accessible via public transport, and proceeded to monitor them since the 1970s to present day. Really amazing. So literally 50 years of data collection. This is the North America's longest running butterfly monitoring effort. In 2019, Chris Halsh and Matt Forrester took over some of the higher elevation sites to kind of take that load down for Art. And I was introduced to the effort in 2022. More recently, Tara Christensen was also brought into the fray, and she's been focusing specifically on some of the plant assemblages that are at these sites, and we'll get to discuss a little bit about what some of her findings were. Our next data set is also very near and dear to my heart, and hopefully some of you are already familiar with this one. It is the North American Butterfly Association, and we have a variety of different sites with different lengths for the data sets over the course of I think it was starting in the 90s is the earliest one up until present day across the entirety of the United States, of course, subsetted down to the Western United States for this particular effort. Pictured here is a Yosemite count. And the premise is you go out to this particular site, walk along a fixed transect, and count every single species of butterfly you see, as well as how many of them there are. And finally, the last one, iNaturalist. Am I admitting? Yes. So this one, I would imagine some of you are also familiar with again, and this is an app that you can download onto your phone, take a photo of any organism that you're interested in, and then ideally, if the photo quality is high enough, we will have a panelist of specialists that look at that individual species and get it down to whatever species it should be. And we can then use these findings within our data set and within our analysis as well. So altogether, we have really excellent temporal coverage. We have great geographic coverage. Our 400 different species of butterfly that were included within this particular work. And with some statistical magic, we then had an overall finding with 96% confidence of 1.6% annual compounding declines. So for every 5 butterflies we saw about 20 years ago, we now on average are seeing around 4. And this feels a little bit hard, but we're going to be talking today about some of those stressors and how we can mitigate them. And again, we do have to remember that these are open, protected spaces where they're probably insulated against any anthropogenic stressors that might be happening within the Central Valley. So things like urbanization, agricultural intensification, light at night, et cetera. So if anything, this estimate might be a little bit conservative as compared to if we had data that were illustrative of areas that have experienced more degradation. Okay, so now getting into some of the drivers, I'm going to start with climate change. So returning to those Shapiro transects specifically, we'll be looking at these high elevation sites first, Castle Peak, Donner Pass, Sierraville, Lane Crossing, and Washington. And this figure here, I know it looks a little bit busy, but we just have our standardized population index on the Y. So anything above a 0 indicates that our populations are doing well, if not a little bit better relative to our historical baselines. And anything under that indicates that our populations are declining in terms of abundance. So these are all color coded by elevation here. So everything, of course, as the high elevation sites. And as we can see, things are primarily stable up until the early 2000s. Again, remembering that these are protected spaces, these butterflies are probably able to ameliorate some of the deteriorating conditions they're experiencing up until this point. So what might have been going on? Well, Matt and his colleagues looked into this further, and as it turns out, minimum nighttime temperatures on average are increasing more relative to the Central Valley. So there's this disparate increase at the high elevation as compared to those lower elevation sites. So rather than being able to cool off, as these are ectotherms, their temperature does depend on the exterior conditions, the ambient conditions. During the nighttime, they're exposed to warming temperatures, so probably a little bit more physiological stress that makes it then more difficult to deal with some of the other things that are going on. Okay, so warming nights led to declines in the mountains, and unfortunately, some natural history traits like body size or generation time per year were not enough to ameliorate those deteriorating conditions. Okay, and zooming out a little bit further, in case you ever find yourself in fisticuffs with somebody who's trying to resist these findings, we can also think about some broad trends across North America. So as we know, things are becoming a little bit hotter on the western side of the United States and getting a little bit cooler on the east. In addition to that, things are also becoming a little bit wetter. Oh, I should explain these figures, apologies. Okay, in the brown, things are becoming more dry. In the blue, of course, things are becoming more wet. So we're seeing disparate trends here where things are drying, increasingly so on the western side of the United States, and conversely becoming more wet on the eastern side. So we would anticipate that butterfly population trends on average would probably be declining within the western United States and conversely increasing in the southeast especially. And that actually is what we found. Crossley et al. Performed this work in 2021, and anything in the red indicates that butterfly abundance is declining, and of course our blue indicates that it is increasing. So those trends are what we see. Okay, now focusing on the pesticide part of the work that I've been involved with, as well as my other colleagues, what is going on in the Central Valley? So you'll notice that beginning in the 90s, we're seeing some pretty substantive declines. And some obvious answers are already available here. We can think about the increase of impermeable surfaces. So obviously this corresponds directly to a reduced presence of butterfly habitat, and accordingly we would see declines, as well as an increase in agricultural intensification surrounding all of these different sites that ART was going to monitor during this time. In addition to this, after Matt investigated a little bit further, these declines also coincided with the introduction of what's called neonicotinoids. And for those who are not familiar, neonicotinoids are systemic pesticides, they're water soluble, and they're also taken up into every cell within the plant's tissues. So beyond just being present within the leaves, they're also going to be found within the nectar, within the pollen, and therefore they're going to be having impacts on non-target organisms. You can think about our cute little butterfly coming to take a drink, trying to restore its energy levels, and instead being impacted negatively by these particular compounds. So they're used both commercially as well as non-commercially in California, and beyond just being found within the plant's tissues, especially when applied indiscriminately, they will probably run off or leach into surface waters, which will have additional negative impacts on things like aquatic arthropods. And historically, these agricultural margins that I've been talking about have been refuges for a number of butterflies. You can think about it in terms of the fact that they're being irrigated for the course of the entire year, so there's not going to be anything that's drying out, which of course corresponds to food availability over the course of the entire year. But in the context of all of these pesticides that we've been seeing so far, is it possible that this is changing? So to answer this question with respect to the monarch specifically, my colleague Chris Hulsh and the Xerces Society, they are a nonprofit for insect conservation, teamed up in 2019 and surveyed a variety of different land use types, including agriculture, refuges, and urban areas for milkweeds, assayed the leaves for a variety of different pesticides. There were 94 different pesticides that were being used consistently within the United States during that time period. And I'm about to show you a very busy graph, but I promise I'll walk you through it. And they found ubiquitous contamination. So here we are looking at the group for each individual compound, so insecticides, fungicides, and herbicides. The rows are each individual compound, and on the columns we have our agriculture, refuge, retail, and urban sites. In addition to that, for each of the squares that is colored, that will indicate the concentration, ranging, of course, from 0 and white all the way to purple, dark purple, indicating 1,000 parts per billion. And now we get to talk about one of my least favorite ecotoxicological standards, which is the LD50. So this refers to the lethal dose that would kill approximately half of the test population. Typically, this is only performed on pest insects and non-target vertebrates because people care about things with spines, and I know that you share this grief with me. But fortunately for the monarch, we do have a number of different values that were established by various studies where people were interested in the conservation of this particular well-known insect. So if we zoom in a little bit further, we'll focus on the insecticides for a moment and zoom in again. These are each neonicotinoids that were discovered in this assay, including clothianidin, dinotefuran, imidacloprid, and thiamethoxam. The only one, fortunately, that exceeded a lethal dose 50 and therefore we would expect to see lethal effects was clothianidin. And this actually brings me to a nice, lovely story amidst all of this pesticide insanity that I'm excited to talk about. So Chris and the Xerces Society were like, oh my gosh, 1,000 parts per billion? That's not really what we would anticipate. They then contacted this grower who was absolutely horrified by this number as he was trying to do his best to ensure that he was using more regenerative practices. He talked with different neighbors as these compounds were not any that he was spraying on his land and discovered that one of his neighbors was spraying somewhat indiscriminately on their property. Fortunately, they worked out a solution and reduced neonicotinoids as a consequence. I find this especially heartwarming because these individuals are working hard, they're trying to create food, and the fact that they have time and compassion for these insects that run the world was really amazing to me. Okay, but back to the pesticide story. Two of the other more commonly found insecticides included chlorantraniliprole as well as methoxyfenozide. And again, these were detected above LD50, so there would be lethal effects. In terms of the breakdown between different sites, agriculture had a total of 11 compounds per leaf. Refuges surprisingly had 7 compounds per leaf, as they are located in an excess of all of this different agriculture. Retail, on the other hand, had 20 compounds per leaf, which was quite surprising. And again, Chris and the Xerces Society were very horrified and wanted to follow up on why this was happening, or where this was, or the extent rather of where this is happening. So they had a variety of amazing volunteers purchase milkweeds from various nurseries throughout the entirety of the United States. They then had the same chemical assay that was performed previously, performed on these leaves to get a sense of what chemicals were present. And tragically, very similar findings. All of the states that they surveyed had some amount of pesticides contained within them. And again, here are concentrations pictured, but we'll just focus on a couple of specific findings here. Initially, they had the hypothesis that store size would be important in terms of predicting whether or not exceedances, that is, there would be lethal effects, would be found within their plants. And unfortunately, there is no relationship between store size and the percentage of plants with exceedances, so it doesn't necessarily matter where you're going. Okay, and then now there's also this label that indicates that it's butterfly friendly. And again, we would have that same expectation that there would be a lower percentage of plants with exceedances for those that have a label. And shockingly, the reverse is actually true. So plants, yes. So plants with a label indicating that they're pollinator friendly on average had higher concentrations of compounds, especially those with exceedances. So a little bit frustrating. And I think the Xerces Society is still trying to establish why this is going on. But of course, supply chains are especially crazy. We don't necessarily know what's happening at each step. So if anything, it might make more sense to be growing things from seed. Okay, in terms of more recent efforts, we were particularly interested in urban areas within California as well as New Mexico. And so we had our amazing team of volunteers sample a variety of different urban parks as well as urban backyards for all of these different known butterfly host plants. So we're expanding just beyond the milkweed purview, and we're going to think about all butterfly species here that are within the area. And of course, we performed the same assay that was performed previously, except the list of pesticides was updated a little bit according to what is going to be used in current times. And today we'll just be focusing primarily on California, as we're pretty close to them. And again, we found fairly ubiquitous contamination. I'd also like to note too that these samples were taken from the backyards of master gardeners who typically are not applying things to their landscapes. So this is probably attributable in part to drift or maybe something their neighbor is applying. So here, rather than individual exceedances for each different compound, we're thinking about many different genera of plants, which are color-coded, apologies, in this legend below, and prevalence is pictured on our y-axis, ranging from 0 to 150, which is the number of plants that we sampled. Each individual compound is listed in small text on the x-axis, apologies, but we will focus on a couple of different compounds here. So 94% of plants were contaminated with at least one and up to 18 different pesticides, somewhat surprising. There were a couple of usual suspects that cropped up again, and these included azoxystrobin, methoxyfenozide, and flopyrum. And methoxyfenozide was detected in 78% of plants, compared to in all plants for Chris's study. In terms of risk for butterflies, unfortunately, we do not have many LD50s available, but for those that were available, we were able to put together an understanding of what might occur if exposure were to happen. So for 28% of those plants containing azoxystrobin concentrations, this would probably lead to a reduction in monarch butterfly wing size, which of course has fitness implications, making it to your ultimate destination, et cetera. And for 14% of plants, those were containing chlorantraniliprole concentrations exceeding known lethal concentrations for the monarch. But I'd like to emphasize, though, for the 26 other compounds we detected, both lethality and toxicity studies have only been done on pests. These are creatures that have been exposed to these compounds very consistently. They probably racked up some amount of pesticide resistance at this point, and therefore their physiology is not illustrative of what we would probably see out in the wild for an array of different butterfly species. Without these values, it's difficult to quantitatively assess the risk to butterflies rather than just saying, we found all of these compounds. It probably isn't good. And therefore, it's difficult to inform policy. So what are we doing moving forward? Well, we recently had a meeting with the California Department of Pesticide Regulation, and they are actually looking into reducing the effects of agricultural spray drift. And this is an area where Angie Leonard, a postdoc in my lab, is potentially going to collaborate with this particular organization. So ideally, we'll see growers being a little bit more conscientious about how they're applying their compounds to ensure that there are limited effects for other non-target species. And that felt pretty good. In terms of quantitatively assessing risk, I will be looking at methoxyfenozide impacts on the painted lady. So currently, as I was saying earlier, we have no numbers in terms of, okay, at this concentration, we would expect these sublethal effects or this lethal effect as well. So after rearing these painted ladies on a variety of different concentrations using Malvaceae, I will be using a flight mill to assess their flight, the impacts of exposure on their flight. And with all of that, I wanted to focus on some good news as well. And I imagine some of you have already seen this in the news. In 2023, AB 162 was enacted, and this bans the non-agricultural use of neonicotinoid pesticides within Nevada. And here is Governor Lombardo doing his thing. Additionally, in 2023, again, a pollinator-friendly plant labeling act was introduced, and this would facilitate a voluntary program wherein growers would be able to send in their plants, have them assayed for pesticides, and then be given a label that indicates to the consumer that they are devoid of pesticides that would have negative impacts on pollinators. Of course, this is currently at a standstill, but my fingers are crossed that maybe at some point this will be passed. That would be amazing. And then finally, in terms of more data points on the map, California will begin tracking seed treatments. So this is just the prophylactic application of a given compound just to prevent pests from being deleterious. Okay, and finally moving into some more fun stuff with flowering plants. If you remember Tara, then I introduced her at the beginning of the talk. She was particularly interested in floral assemblages, and she set up her surveys to mirror roughly the time frame in which we were visiting our different sites for butterfly monitoring. She included a range of 10 to 12 plots at each site, covering a diverse number of different habitat types. And within each plot, she identified every individual flower all the way down to species, and then proceeded to count all of the inflorescences to give us an understanding of how much nectar was available for butterflies. So tons and tons of work. She told me she would dream about flowers and counting them. And she was particularly curious about whether or not butterfly diversity depended on floral abundance or floral diversity, as well as how these two taxa are being impacted by weather. And I'll show you some of a snippet of her preliminary results here. So we're going to be walking through what's called a Bayesian hierarchical model, and it's basically just a fancy way of saying we're looking at causal relationships here. So our predictors include flower abundance, minimum temperature, precipitation, and day as a polynomial term. And of course, we're interested in the impacts on flower richness as well as butterfly richness. And I should clarify when I'm saying that, I mean just the total number of species here. In terms of day, which makes sense, as day increases, so does flower richness as well as butterfly richness. Similarly, we see the same for precipitation. And finally, and excitingly, in my opinion, an increase in flower abundance leads to a direct increase in flower richness, which of course makes sense, but this also has a direct impact on butterfly richness as well. Whereas minimum temperature, which we would have expected to influence butterfly richness to some extent, did not have any impact. So butterfly diversity within this area is actually strongly related to flower diversity as opposed to proximate weather. I thought that was very fun. In terms of phenological mismatches, although her work was conducted over 3 years, we have a sense that things might not necessarily be matching up every year. In 2022, so we're looking at our date of peak abundance here at the very top, and then our x-axis is date of the year. Butterflies are on the top here, and flowers are on the bottom. For 2022, things had a very similar peak, which was great. In 2023, there was an 8-day delay for flowers as opposed to a 17-day delay for butterflies. And finally, in 2024, again, a bit of a mismatch was occurring where there was a 15-day difference for flowers and a 21-day difference for butterflies. So although we can't say anything conclusive as this was only done over 3 years as opposed to the time span on which these things happen, there are implications for phenological mismatch here. All right, so bringing it back to some efforts to facilitate conservation as well as what we can do on the ground. Matt was particularly interested in which species and groups have experienced the worst decline and whether or not we can leverage the collection of data as well as some conservation efforts to mitigate those impacts. So here we're looking at an extremely busy graph, but this is just a phylogeny of butterflies, so how things are all related to each other. And they're color-coded according to the group into which they fall. Our cute little skippers are in yellow there. The dark blue includes our Papilionids, so Western tiger swallowtail pictured in the bottom right. Red would be our nymphalids, so things like monarchs, painted ladies. Blue butterflies or the lycaenids. Dark red is Rhyodinidae. And then finally, green for Pierids, otherwise known as the whites there. Risk is indicated by the hue of purple. Low is indicated by a nice light lilac, and a dark purple indicates a high risk. And they found that no one trait was perfectly predictive of decline, and they were considering a variety of different natural history traits, such as host plant range, the range or geographic extent in which the butterfly is found, the number of generations per year, as well as wingspan. But broadly, if we look at the concentration of risk distributed over this phylogeny, smaller bodied species tended on average to have a larger proportion of risk. So for our cute little lycaenids here, as well as our skippers, they're doing a little bit worse. So let's see some of these faces that are having a hard time right now. These include the skipper up here, Mexican cloudy wing, the tailed copper to the right, and finally, one of my absolute personal favorites, Haberdeus gruenus, or the golden hair streak. And to tell you a brief little story about them, they emerge in the early summer, and then they don't reemerge until the entire end of that particular season, so in autumn. There has never been one single record of them feeding on a single plant, and it's believed that they are actually feeding on the exudate from, or the honeydew rather, of aphids. So if you ever see this in the wild, please take a video. That will be the first instance of that ever being recorded. I would love to do that, but it's a lot of time, I think, wandering around in trees. Okay, there's a little aphid. In terms of our top decliner, this would be Vanessa Annabella, otherwise known as the West Coast lady. We suspect that the loss of the second generation for any number of different reasons is probably why they're on the decline. Previously, they were very, very abundant at sites like Sassoon Marsh within the Central Valley. And now I'm quite excited to see one of them if I go out. Okay, but that's a lot of doom and gloom. And I think it's important to focus on things that are faring well as well. And pictured here is one that is actually having a great time in the context of all of these anthropogenic stressors, the Western tiger swallowtail. So they feed on aspens and cottonwoods. They lay their eggs singly on the top of these leaves. Our cute little larvae will emerge, continue to feed, and develop in a silken mat, and they'll overwinter as a chrysalid. You'll see these emerge generally in June and July, and that's when they'll be on the wing, typically floating around their host plant. So Matt and I were especially curious as to what their host plant usage looks like, as well as what they're feeding on when they're adults. And fortunately for me, especially in the context of migratory species, these miniaturized trackers have finally become available for tracking butterflies. So what we're looking at here is a miniaturized solar panel that's going to be powering the entirety of the device, as well as this receptor that's going to be pinging your Bluetooth network. So if your phone is within 30 to 100 meters of this receptor, it will upload this information to the cloud, and we'll have a general understanding of where that butterfly is on the landscape. So if you see any of these floating around in June and July, don't panic. It's not the government. It was probably myself or Matt. And so again, just a little vision of what we're hoping to get a sense of. Where are they moving throughout Reno? Are they navigating through this nexus of houses and urban spaces in a particular way? Are they just booking it over houses? What are they eating? Are those things contaminated with pesticides? But it just doesn't matter because they're so large. We have so many questions. Okay, and moving on to some local efforts that we can all undertake. I would imagine these are all things that you guys are speaking and thinking about. So apologies if this is a little bit redundant, but we can also make our backyards a habitable space by reducing mowing for pals like our cute little skipper here. Typically, these grass feeders will lay eggs singly on the top of grass stalks. So if you're mowing anything, obviously our cute little eggs are going to be mashed by those blades. Additionally, planting or leaving host plants, and I know this is a bit of a big ask as I am currently at war with a bunch of Malvaceae in my backyard, and it's become very woody. If you don't have time to get around to it, that will just mean that you'll have a bunch of checkered skippers in your yard this spring. So for me, that will be the case. Additionally, bumblebees also like malvaceous plants, so they're not all bad, even if they are encroaching on your beautiful space. Finally, if you are going to be applying pesticides, you can reduce your use of them or use as directed to ensure that there aren't any non-target effects. And additionally, if you are curious about the pesticides and the active ingredients that you're using, you can use the Ecotox knowledge base to look up your compound and find the whole array of studies that have been conducted on that particular chemical, and just get a sense of how it impacts non-target species. And finally, the lovely Cynthia Scholl, who is in charge of outreach here at UNR, has put together this wonderful list of host plant and larval associations, as well as where you can find seeds close to us. And you can think about host plant phenology, that is, when are things flowering, to ensure that butterflies and other pollinators have access to food over the course of the entire summer. And pictured here are a couple of our local natives, excluding that chokeberry. I messed up, it's not native, this one particularly, so ignore that. And finally, if you have the time, using iNaturalist is yet another great way to help out scientists. If not myself and you're taking a photo of flowers, I know that somebody out there will be immensely grateful to have that data point. And then with that, here are my resources. If you're curious about them, you can send me an email. And thanks so much for your attention tonight, guys.
Thank you very much. That was fascinating. Quick question. Do you guys look at the plant, some of the plant iNaturalist things in the areas where you're tracking the butterflies? So you are looking at plants as well as
I'm still getting a better sense of host plants and which species are which. But yeah, that is important. Yes.
Right. Does anyone, if anyone here has questions, if you want to come to the front. Okay, how do I want to stop sharing the screen, don't I? So, okay.
Yep, stop sharing.
So if people have got, okay, click that. I'm not very good at that stuff. Okay, if any, let's see. Oh, there's one question in the chat. So if anyone has any questions, oh, we'll start in 5 minutes. If anyone has any questions, anyone online, please put them in the chat. Does anyone here have any questions? Can you come up to the front? Because that way the people online can come in here too. Thank you, Claire. That's fascinating.
Thanks for having me.
Oh, no, we appreciate it because it's all interlinked. I mean, and you have the problem. I mean, everyone knows about monarchs, but they don't know about the other ones. Right. And, you know, people know about some of the garden flowers, but they don't necessarily know the native flowers, just trying to get the word out. Anyway, if you'd like to ask your question, come and stand where you can be.
Right, if they can hear me. Great talk, by the way. That was really cool.
Thanks.
You mentioned the Malvaceae that those skippers like, and you're showing the cheese mallow. But there's like the native Malvaceae of like Straussea.
Oh.
Do you know if the skippers will go on those, or like do you know if a lot of those plants that go on
I think
generally
the entire genus is available to them. So if you have a local Malvaceae that please use that one instead of whatever's going on in my backyard.
Yeah, okay.
Okay.
If you ever want a Straussia, I can give
Yeah,
you one.
that would be awesome. Thank
Okay.
you.
I just have a general comment.
Can you, yeah, sorry. You've got to work out how to share those things.
Hi, my name's Jackie. Having lived in this area for over 50 years and seeing what's going on, one thing I know in talking to people that are farming for a living, chemicals are really, really expensive. So if you're having a dialogue with anybody, whether it's your neighbor or a farmer or somebody, to point out that they're wasting their money by overspraying or spraying on a windy day and everything, that might get through where something else doesn't get through.
It's a great point.
Okay, it's just an
It's what the
idea.
others- yeah.
It's one more thing to have in your lexicon of communication. Really expensive.
Nancy, I will get those links and write a little blog post on the website, which means it'll stay up there for a while, and share them on the website, so you don't have to wait 2 months till the next newsletter. Any other questions? Any other questions online? Marcus, do you have any questions down there?
I did have one question. So when you had the map of the North American Butterfly Association and all those little transects, those were like set, those don't like shift from year to year, right? It's the same location.
No, it's the same location every single time. So for each dot that corresponded to one site, it would be that same exact transect every single year.
Okay. I mean, it just seemed, Nevada was very underrepresented. I noticed on that.
We do have our Toyabe count, but if somebody wanted to take on an additional count and start that, we would love to have that happen.
Yeah, I was just wondering about like if it was, if it was possible to add more, but it sounds like it is.
Yes, yeah, I imagine that NABA would be very excited to hear from anybody about having an organized effort or group of volunteers to go out every single year.
Perfect.
Okay, thank you very much. Well, thank you to everyone who attended, both in the room here, online, and in the room down south. Our next meeting will be the 5th of March, and I don't know what the title is.